Hi there, everybody. I'm really excited to be talking to all of you today. It's really a privilege. I'll be talking today about accurate cross-species out-of-distribution predictions of protein-protein interactions using deep learning, which really rolls off the tongue. A brief introduction as to who I am. I'm Joseph Szymborski. I'm a PhD student at McGill at the Electrical Engineering Department. My supervisor is Amin Ahmad. I'm part of the Combine Lab there. I'm also part of MILA, the McGill, the Quebec, it would make more sense if it was McGill, the Quebec AI Institute. These are my, find me online at Mastodon or my personal website. So let me begin into a brief introduction to the problem we're trying to solve. I'm primarily concerned with protein-protein interactions, or PPIs. And most of what cells do, I'm surprised, I feel silly saying this to this room, but most of what cells do is in fact achieved by these physical protein interactions. And when proteins interact, they can make chemical or physical modifications to one another. And to the delight of computer scientists in the room, you could think of this as a computational graph where each protein is a node and each interaction is an edge. And that's an incomplete model, but historically it's gotten us really far. So the question then becomes, given two proteins, how do we determine whether they interact? Historically, the way that we do this is we go to the lab, we go to the bench, and we perform co-immunoprecipitations, for example. And these wet lab experiments have the characteristics that they're slow and that they're expensive, but they're evidence. You have confidence that you're witnessing two proteins interact. The consequence of this being slow and expensive is that we can't really comprehensively and exhaustively search every single protein interaction possible, all pairwise, and especially not across all known organisms. And the result of this is that we have some organisms that are really well studied and that we have a lot of PPI data for, and some that we just don't for various reasons. They're hard to work with, or we're just not particularly interested in them, or nobody bothered to ask. And this is a dangerous audience to start, mentioning, facts about string. But the string database is a comprehensive database of protein-protein interactions. And if you look really closely, you'll find that actually at the class rank and at the order rank, taxa are actually really not well represented. It's not a uniform distribution by any means. Only 30% of taxa at the class rank are represented, and only 16 at the order rank. So we have a lot of PPIs, a lot of organisms with little to no PPI data. So another way to predict protein-protein interactions is to use computational models. And they have great properties. They're fast and they're cheap, but it's not evidence. We're making a guess. We're making a prediction that these two proteins interact. But these two properties, fast and cheap, perhaps put them forward as, you can perhaps posit that they're the answer to this species gap problem. Maybe that's how we bridge it. So there's a small problem with that. It's that good ML models, good machine learning models, run on lots of good data, garbage in, garbage out. And these non-model species don't have a lot of data to train on, or any data to train on. So you can't really create models that do a good job. So what we need is something that's able to do cross-species. So instead of doing intra-species predictions where you're training on human and testing on human, you can do something where you train on a well-understood organism like human and then test on one that is less studied. But to do that, you need to have a model that's capable of making out-of-distribution predictions, that is able to generalize to other species. And historically, we have a problem with that. And that's been known since 2012. There's this great paper that, if you've ever spoken to me about, I'll go on and on about, by Park and Marcotte, where they study this phenomena. Basically, what happens is if you do like most papers did, and frankly still do, and you have a bunch of PPI and you want to train a model to make predictions, what you'll do is you'll commonly randomly split these interactions into training and testing. And Park and Marcotte found that with that evaluation scheme, methods performed quite well across the board. What you're missing there is AUROCs of all these methods doing really well. But they went forward and said, okay, well, what happens if you call some of these proteins training proteins and some of these proteins testing proteins, and then you eliminate all the interactions between the proteins that go between the types, so only training and testing and testing. And what you find is that these methods that did so wonderfully, all of a sudden their AUROC falls to basically random. And this is a problem, even though that paper was written in 2012. There's a great paper that's being presented tomorrow at ISMB. I only found out now. But there's this great paper that has great stats on this, that even today in 2023, we're still doing a lot of random sampling, and a lot of our performance is still being attributed to this. So definitely go check out that talk, Cracking the Black Box. So there is a problem, right? It's that species gap, and that we can't generalize to do these cross-species interaction predictions that we need to do to bridge it. So what's the solution? So obviously I'm going to say it's my method. Rapid is actually a method that we presented last year at ISMB, but we've never really looked at cross-species and cross-species performance. So if you're not familiar with Rapid, what it does is it's a sequence-based method where it takes sequences, amino acid sequences, it chunks those amino acids into tokens using a sentence piece tokenizer. It then goes through a deep network AWD LSTM encoder that creates fixed length vectors that are representations of those sequences. We concatenate those latent vectors, or at least we did in the original paper. Now we multiply them, but and then that goes to a fully classified, fully connected classifier layer. That's just a two-layer fully connected network, and that creates interaction probabilities, or it generates. And you know, this isn't the only twin neural network model that does PPI prediction. But we do things that are different, I swear. There's lots of regularization, and you know, there's a lot of little details that make us very different. I suggest you read the paper if you want to know it. But I really don't want to talk too much about architecture today. I do want to talk a bit about the data, because I never thought that I'd spend so much time yak shaving on data processing. It's so much. It took so much work. Part of that was splitting the proteins and edges the way that I described. Part of it was making sure that protein sequences between training and testing proteins weren't too similar. They weren't more than 90% similar between training and testing. So that way there's no accident. We call them two different proteins, but really they're not really different. And we did that using UniRef 90. And we sampled negative pairs randomly. And we did this based on a wealth of literature that describes that it really is the best way to do it without really biasing your data any further. But less on that, come talk to me later if you have thoughts on that. So what we did was we created a convenient tool, because I had to keep on making these data sets for different species and do them for different random seeds. So we actually created a tool to download and process third-party data sets like string and UniRef 90, make new robust data sets for this PPI. So this command will create a data set like I described. And then you can actually convert it into different formats that are ready for other PPI programs like Descript and DPPI and Piper. But that was about the data. Now let's talk about the performance and how we actually did. So it turns out that rapid is more accurate than leading methods. This is something that we were able to show last time with our last paper, but we've added some new methods here. And actually our data sets are a little bit different. They're a little more strict. We've controlled for more things like that sequence similarity. That's not something that we did in our previous paper. And we find that rapid outperforms other leading methods, 0.767 AUROC on the testing set for a human benchmark. And what you'll see here in red, I have Descript, which is the second leading method. And I noted in red because it does contain non-human data. So Descript leverages a protein language model, a large language model. And that language model is trained on basically all of UniProt. And that will include proteins that are excluded in my strict data set in that testing set. And we'll see other ways in which these large language models that are becoming very popular can actually contaminate and leak data. So it's important to pay attention to that. We're also really excited to show that, and this was an experiment that was inspired by that Cracking Black Box paper. The idea is that if you have a data set, a PPI network, these tend to be scale free and have really weird degree distributions. And so if your PPI method, your PPI prediction method is only learning the degree distribution and outputting those, then if you have a scrambled data set where the IDs of the proteins are all messed up, then it'll still think it's doing well, even though there's no biological data there. So it's not really learning biology. So we want to see how Rapid would do. If it learns biological features, it'll do well on string, and if it doesn't, it'll do poorly on the scrambled data set. And that's exactly what we saw. We're really happy about that. Another thing that we were really happy about was that Rapid performs well across species on the cross-species task. So this graph illustrates that Rapid, when it is trained on human PPIs, maintains its performance when tested on other evolutionarily distinct species. So on the y-axis, we have that Matthews correlation coefficient that measures our performance. And on the x-axis, we have that evolutionary distance, so the evolutionary distance from Homo sapiens. The unfilled markers here represent the performance of Rapid that Rapid achieves when trained and tested on the species indicated by the color. So that's intraspecies. The filled markers is the cross-species performance. And so what we can see is that for most cases, except for maybe the yeast at the far end there, we're doing really, really close to that intraspecies performance. And we could also see that if we compare this to other methods, we actually beat out most of the other methods on all the organisms except for C. elegans. I will note that it only is close between Rapid and Descript, which again uses all of Uniprot. So it'll include other species as well in that large language model. So it isn't exactly a apples to apples comparison when it comes to this test. Another thing that I really wanted to see was I really wanted to visualize this out of distribution challenge that we were facing. And this is not the perfect analysis, but it really it's an intuitive one, which I love. So basically, what I did was I encoded sequences, all these sequences from Uniprot, and I one-hot encoded them. Then I mapped them with UMAP into 2 dimensions. And when you can see them here, black is human in every case, and then you have different species that are further in evolutionary distance as you go along. And what you can see is that these are completely different distributions, right? These are distinct clusters. So I wanted to see what happens when you encode Rapid, when you code these sequences with Rapid and you get those latent vectors. How does that look? And what you see is they start to integrate much better than they did before. This is one of those things where if you look at a UMAP, you see what you want to see. So we wanted to quantify these effects, and a nice scatter plot like this is nice, but it's nice to have a little bit more of a quantitative measure. So if you're familiar with scRNA sequencing batch correction techniques, you may be familiar with LISI. It's the local inverse Simpson's index. Long story short, it's a way to quantify how well integrated data points can be that are labeled differently. And so what we see here is the LISI scores. 2 means that it's as integrated as it can be. 1 means it's as not integrated as it can be. And what we see here is, for example, the first box plot is how well mouse is integrated with human. Second is with zebrafish and human, and that goes on. And you can see as evolutionary distance goes on, that LISI metric drops a bit, but it's still much higher than it was that one-hot encoding. So it is very interesting. I want to pause for a moment and call back to our architecture. So this is what it looks like. A comment we get all the time is, what happens if you replace the LSTM with one of those protein language models? What if you add attention? What if you do the cool thing? And we kind of did that in the original Rapid paper, but we never used one of those pre-trained methods like ProteinBERT, for example. That was actually presented at ISMB, the presentation before mine last year. So we thought the same thing. We figured that was a great suggestion. So we did that. We stuck ProteinBERT into Rapid. So the encoder goes away, and we just put in this large protein language model, and we ran it as that. And now this is a reminder that all those things I was saying about Descript now applies to this new RapidBERT model, right? So when you add this language model, suddenly you have all this knowledge from all of Uniprot from all those species. So there is threat of data leakage there. So when we test this RapidBERT, Rapid still outperforms the other methods. But it's a dead heat, frankly, but only between those methods that sort of are not on the same wavelength, if you will. And again, RapidBERT, it's a bit of a different story with the cross species. There we start to trade blows more with Rapid, where RapidBERT and Rapid both basically it's a dead heat between the both of them for this task. So one thing that I'm really excited about and also really nervous about is that this is the first time I'm speaking publicly about rapid.org. So rapid.org is an online version of Rapid. It is really early. There will be bugs, and I promise you, that we're going to try to get this up and running as fast as possible. But you can visit it now and make whole proteome predictions. So you put up an amino acid sequence, and it will spit out all the interaction probabilities between between that amino acid sequence and all the other sequences in UniProt for Homo sapiens. Right now, it's only Homo sapiens. How long will it take? So you'll wait in queue, depending on how many people are asking for it at the same time. It'll process 2 at the same time. And my test is taking about 6 minutes. So that's the sort of order of magnitude you'd expect. And then you get whole proteome predictions. So hopefully I don't know, if the site crashes, we won't earn our name. If it takes 6 minutes, I think we earn our name for Rapid. So in conclusion, there is a species gap. We need models capable of cross-species prediction to bridge that species gap. We need generalized models to conduct accurate cross-species predictions. Rapid is a generalized model that outperforms leading methods. Rapid makes accurate cross-species predictions. Rapid predictions are based on biology, not network topology. And Rapid is computationally efficient. And these preliminary results show that we can actually launch a web service and make this public to everybody running on one very small CPU server. So I'd love to thank my lab, my supervisor, Amin Ahmad, and all our supporters. And again, if you want to talk about Rapid afterwards, I'll be at my poster, A394, or you can reach me online. So thanks so much.
All right. Again, we are a bit short of time, but let's see if there is maybe one question to answer. Yes, okay. Mm-hmm.
Thank you, brilliant work. Is this is there a command line, like, can you run this on the command line?
You can run Rapid on the command line. Yeah, it's frankly, it's not a super usable experience at the moment, but it exists and you can run it. Yeah.
And you said the rap.org predicts any protein sequence, interactions of any protein to all human proteome.
Exactly. You put any amino acid sequence, put it in the box, and hopefully it puts out those sequences. Again, we haven't done a lot of validation, so we don't know, if it's as valid as the command line version is that we've validated super well. But yes, there's a newsletter on rapid.org. Sign up, and we'll let you know when everything gets a green light and that we're super confident about our results, and there's more organisms and things like that.